The second way that bacteria increase their genetic diversity or exchange genetic material is by conjugation. So conjugation is a physical connection between two bacterial cells. Essentially, it's a tube that allows the transfer of DNA from a donor to a recipient cell. So, conjugation involves direct contact. Transformation, that we talked about in the last video, does not require contact. The cell simply takes up the DNA from the environment. Conjugation, however, requires a physical connection. So it occurs commonly in many bacterial species. Generally, it occurs between closely related species or between two bacterium that are the same species, including E. coli. So conjugation requires something called a fertility factor. Now, a fertility factor encodes the genes that are going to make this connection. So this fertility factor encodes genes that are going to make what we call a pilus. So a pilus, or pili for plural, from the cell with the fertility factor is actually going to form and grow between one bacterial cell and the other. So the cell that has this fertility factor is called the F positive cell. And that F positive cell is going to be our donor cell because it encodes the genes that are going to make this connection. And then the DNA from that donor cell is going to be transferred into the bacterial cell that it is transferring DNA to. So that other cell is called the recipient, and generally, or they don't have to be, but they might be fertility negative, which means they don't encode the gene for this fertility factor or this ability to produce this connection or this pilus. So this transfer of DNA, once the pilus forms, happens via something called a rolling circle model. And we'll talk about it a little bit more in a couple of slides. There's a nice little picture, but essentially the DNA, a little nick happens in the DNA, and at that nick, a single strand sort of unwinds and is passed through to the other cell. Once in the other cell, it is turned into a double-stranded DNA molecule and maintained as a plasmid, or it can also be incorporated into the genome. So once this F plus cell, or this cell with the fertility factor, infects the other cell and transfers it, this new cell is now F positive. It now contains the genes for the fertility factor and any other genes that are encoded on this plasmid. Now we'll see in a few minutes, this fertility factor can also be encoded in the genome. All right, so prior to transfer of DNA, once the pilus is formed, so first step is pilus formation. And again, those genes are encoded in the fertility factor. There's a set of genes on your plasmid. Once the pilus is formed and prior to transfer, the plasmid is nicked. Nicked means cut at what we call the origin of transfer, or the ORI-T site. Now, this is a specific sequence within the plasmid that is going to start the transfer of that strand. So this nicked strand is basically going to unzip from the plasmid and be fed into the next cell. So as this plasmid sort of unrolls and unwinds, it is transferred to the donor cell. Once in the donor cell, replication begins to turn it back into a double-stranded plasmid. And then in the donor cell, that plasmid, the remaining half of the plasmid, is then replicated back into a double-stranded DNA plasmid. So once our DNA strand is nicked at the ORET site, the origin of transformation, the origin of transfer, sorry, not transformation, it is going to unwind and essentially be thread through that pilus from our F positive cell to our recipient, our F negative cell. And that single-stranded piece of DNA is going to be transferred into the new cell. Now you can ignore this HFR for a minute. We'll talk about that in just a few minutes. For now, we're going to pretend that this is a plasmid. And we've also got our genomic DNA over there. So we'll just pretend that this is a plasmid that has the fertility factor. It is being unwound, transferred through to the other cell. And now that this gene, this fertility factor, is in that cell, this one is going to become F positive as well. And that means that it can conjugate with another bacterium. It can perform conjugation and continue to share its DNA. Sometimes this fertility factor can actually be integrated into the genomic DNA of the cell. So this F plasmid, these plasmids that encode that fertility factor are called F plasmids, and they can actually integrate into the genome. Now remember in our last video, we talked about how integration occurs. It occurs as a double crossover with the cell's genomic DNA. So generally integration happens at a specific site, and once this fertility factor is transferred into the new cell, it can be maintained as a plasmid in the new F positive cell, or it can integrate into that chromosome. When or if it integrates into the genomic chromosome of that bacterium, it becomes what we call an HFR cell. HFR stands for high frequency transfer. So this cell now has encoded in its genome the ability to share DNA with other cells in its environment. So generally this happens at a specific site where the sequence of this fertility factor is similar to a region in the host genome, which is why this generally occurs among related organisms, related species. It can, though, integrate into other parts of the chromosome, and that's what these lower cells are showing. So in our last video, we talked a little bit about recombination and integration of this foreign DNA into the genome, but we didn't talk too specifically about it. So let's talk about how this fertility factor or how this fertility plasmid can actually integrate into the chromosome. So as I said, integration occurs as a double crossover between homologous sequences that are present in the plasmid and the host genome. So the most common site of integration is between the 3A and LU genes. So the 3A gene and LU gene refer to threonine and leucine. These are amino acids, and these are going to be genes that synthesize these amino acids. So there's a sequence similarity between the sequences, the DNA sequence between this 3A and LU gene that is similar with a region on the fertility factor plasmid. So these regions, because there is sequence similarity, are going to join. Because they are homologous, they're going to line up together, and a double crossover is going to occur. Now, when that double crossover actually occurs, you get integration. This isn't really drawn to scale. You get integration between those two amino acid genes, and now you have the plasmid with the fertility factor encoded in the genome. So now this genome contains, the HFR plasmid region. So now this genomic sequence has the ability to share DNA. So really this slide is just a summary of this HFR cell transfer. So when that the F positive plasmid is incorporated, it converts this F negative cell into a cell that is now HFR. And so the important thing here is that this gives the cell the ability to transfer genomic DNA via conjugation. So this cell here can now conjugate with another cell and transfer his DNA into that cell. So it allows for this continuous passage of genes from one cell to another. One of the applications of this HFR cell is mapping. So we talked about mapping in chapter 9, and I very quickly talked about the E. coli or bacterial chromosome that are mapped in minutes. This comes from the ability of cells to conjugate and transfer their DNA. So the first genetic map of E. coli used conjugation in order to determine the location in quote-unquote minutes rather than absolutes, in minutes based on how long cells were allowed to conjugate. And so let's say we have an E. coli with its genomic DNA, and it is HFR. So it has the ability to transfer its DNA to another bacterium. And let's say that it produces a pilus and it begins to transfer its DNA. Now, generally this happens quite quickly, but if you interrupt it, if for some reason conjugation is interrupted, you're not going to get a full transfer of the chromosome. And if this pilus is quite fragile. And so if you allow these cells to conjugate for a set period of time and then disrupt it by mixing up the solution that they're in or somehow separating the cells, you're only going to get a partial transfer of DNA. So if you do this repeatedly and interrupt at specific time intervals, you can monitor how much of the chromosome has been transferred from one to the other. Now, the part of the chromosome that is closest to the origin is going to transfer first. So any cells that are allowed to conjugate for a short amount of time are going to get the first genes that are closest to that ORET site. The longer that you let it run, the more genes you are going to find have been transferred. So the order of the gene transfer, basically if you monitor the genes that are transferred at time intervals, is going to correspond with the arrangement around the loop. And then the time between gene transfers is going to give you a relative distance. So this first map of E. coli was a relative map, just like we had our relative maps based on recombination frequencies in eukaryotic chromosomes. We also can get this relative map in minutes for the E. coli chromosome. So if our ORET, our origin of transfer, is here, and we allow this E. coli to conjugate with another, it's going to start transferring its genes. Now the first one that's going to get transferred is this 3A gene because it's closest. So when this opens up and is transferred, it's going to transfer the 3A gene. If we let it continue out to 2 minutes, our cells are going to have the 3A and the LU gene. If we continue out to 6 minutes, we get the pro-A gene, followed by the lac gene. So the longer you let this go, the more genes are going to be transferred. Now you can use this to determine the map order of a circular chromosome. Because if you interrupt it, let's say it's 17 minutes or just before 17 minutes, all cells are going to have these first 5 genes. If you let it go longer, they're going to have all 5 plus the gal gene. And so you can interrupt it at specific times and determine what genes come next. Now, this is a tedious process because you have to, every time you interrupt it, grow it and look for the genes. Basically, these are probably all some sort of nutritional mutant. They have some way, or some phenotypic mutant. You have some way to determine that these genes are present. So let's turn this into a problem. So let's say we want to map a bacterial chromosome and we let conjugation happen. And after a certain time period, we stop it and we look to see what gene is present. And when we stop it at that particular time, 90% of our cells have XYL plus gene. So that means that the first gene transferred is going to be that XYL plus gene. Now we also notice at this particular time, 80% of them have the MAL gene. So this MAL gene is going to be the next one because not all of them have transferred it yet. If all or most have transferred the XYL gene, we know that that's early on. If less have transferred the MAL gene, we know that that has to come next. So let's let it run, at the same time interval, a few of them have histidine, the His gene. So this means that at whatever time interval we've had, the order is related to the percentage of cells that have that new gene. So if we look at this specific time, none of them have the LU gene yet. But if we let it run a little bit longer, let's say now 100% have the XYL, 90% have the MAL gene, 30% have the His gene, and now 10% have the LU gene. We know that that one is next. So at a specific point of infection, you can analyze, determine which cells have what genes, and therefore determine the order. So our gene order is going to be XYL, MAL, His, and LU. So if we put this on a circular chromosome and we label our ORET site, the first gene after that is going to be the XYL. Close to that, because most of them have it, is the MAL gene. Far away, because only 20% have it, is going to be the His. And then I just happen to call it 10%. We'll say that the LU gene is close. Now, this is why we said it's relative, because these don't necessarily correspond to a specific distance, but we do know that these two are close and these two are close, and there's a large gap in between. The more genes you have, the better you can fix or determine your actual relative map distances. So here's another way that we might be able to look at our gene order, determine the specific order on our chromosome. I'm sorry for these letters here. I'm not sure how to get rid of them. They're not disappearing when I try. All right, so let's say that we are given some specific information, and I've shown it 2 ways here, and we are asked to determine the gene order. Now, this is not at all difficult. You're probably like, oh my gosh, I've already got it. And you probably do already have it. So if we let conjugation happen for 10 minutes and then interrupt it, and the gene that we see, AZI, that's going to be our first gene. So if we're doing our gene order, we've got AZI. If we let it go, run another experiment, let it go for 15 minutes, interrupt. Now we have the TON gene. 20 minutes, now we have the lac gene. Then we have the gal gene. So you can see that this is actually quite simple to determine. Those that are transferred early on are going to be first in order. Over here, this is like the previous problem. After 10 minutes, we have roughly 10% of our cells with the AZI gene. After 15 minutes, we've got about 50% of our cells with the AZI gene, but now we also see about 15% of our cells have the TON gene. At 20 minutes, we're getting up to about 80% have AZI, 40% have TON, 20% have LAC. So again, we can see our order here. Now, keep in mind that this is a circular piece of DNA. So if we've got AZI, TON, LAC, and gal, this is a circle. And at any point in the circle, let's say we have some other genes here, any point in the circle could actually be our origin of replication. So let's say our origin of replication is actually here, and we've got these other genes behind it. If our origin of replication happens to excuse me, start here before the A gene, we're actually going to get a different order. We're going to get A, B, C, D, E, and then our AZI, then our TON, then our LAC, then our gal. If our ORI site is here, we're going to get a different order. We're going to get D, E, AZI, TON, LAC, gal, A, B, C. Now, if you look at this, we still have the same order. We just have a different origin. So we can also use the order of transfer to determine their location around the chromosome. So let's say we use this interrupted mating technique on some HFR bacteria, and we determine that there are several different orders. Now, there are different origin sites, or there are different ORI-T sites, and each of these ORI-T sites is different for each of these strains of bacteria, but they have the same genes, and they're going to be in the same order. So we can look at this sequence, determine where they are overlapping, and actually determine our gene order. So I am going to start with number 2. It actually doesn't matter, but you'll see why I'm going to start with number 2 in just a minute. And I'm going to rewrite this, and we are using this information, the sequence information, to determine order. So now I'm just going to determine where things overlap. So if I look at strain 3, I see we've got some overlap here. Okay, so UMBACT overlaps with CTERIU. Now, it did not matter whatsoever what I started with, but I started with those two because I know the answer, and you might actually be able to see already what this is going to spell. This is just for illustrative purposes. If you look at these other strains, 1 and 4, you'll notice they are, oh, I'm sorry, actually 1 is not. Let's add in 1. So now we've got this overlap, UMBACTERIUMB. Strain 4, I got a little ahead of myself. Strain 4 is actually backwards. We've got the same sequence, but it's backwards. So this one has transferred the other way. This one, if we turn it around, BACTER. So we can get our full order by sort of adding these together. Our gene order is actually going to be BACTERIUM. Now, the reason that this happens, that you have these other combinations or other sequences, not other sequences, but other origins, is that when we overlap our sequence, we begin to see the circle. So if this is our origin here, we map our genes around the circle, we can see how if we change our origin, we can get other sequences. So if our origin is here, this corresponds to HFR1. Our origin is here, the C, that corresponds to 2. 3, I'm sorry, that was 3. 3 is, oh goodness, I got these all mixed up. 2 is here, sorry. 1 is here, ERI, and then 4 goes the other direction, but we get BAC, sorry, is the other direction, RETCAB. I confused myself because I turned it around. But I hope you can see now this overlapping circle model and how you can use the order of transfer to determine the arrangement around the circle.